Okay, so we're going to talk about amino acids. There is a naturally occurring amino acid, and naturally occurring amino acids all have this carbon, which is sometimes this central carbon, sometimes called the alpha carbon, and attached to that we have a carboxylic acid group. That's where the acid bit comes from. We have an amine group. That's where the amino bit comes from. We have our hydrogen atom, always in the 20 different naturally occurring ones, and then we have this variable bit called R. So you can see that that central carbon atom is going to be asymmetric for all of the naturally occurring acids. Asymmetric. The only time when it's not is when the simplest thing that can be, the R group, is in the amino acid glycine, and that is a hydrogen. That won't have an asymmetric carbon atom, but all the other 20 do. So the next simplest thing it can be after hydrogen, for example, is the methyl group. That's called alanine, and you can see that that carbon there is asymmetric. So there are two possible optical isomers of alanine. As you will guess, that there is only one isomer which is ever found in nature, because enzymes, of course, because the amino acids have to fit into the active site, they only ever form only one version will work, and that it's the same version in from primates, from human beings to plants to E. coli to bacteria. Everything use the same version, the same optical isomer of each amino acid. Okay, let's get rid of that. Now we need to talk about the acid-base properties of amino acids, right? Because you've got two groups there, and they can change depending on the pH. So, for example, this so the carboxylic acid group has the tendency to lose the proton and become negatively charged, and the NH2 group is fairly basic, and that has a tendency to accept a proton and become that. Okay, you've got double charge in there, and that's actually called the zwitterion. We'll talk about a bit more about that. But what is interesting is that you'll see, and this is really quite important, is you'll see, of course, amino acids, you'll see their formula written like that. And they can never exist like that. They will never exist like that, because if the pH is neutral, then the carboxylic acid group will exist as will be negatively charged, will be ionized, and the amine group will also be protonated. Okay, so you can never have it. So let's think about, say if you had these two, if you had this species here. Okay, you've got two acidic species there, two acidic groups. You've got this one has a tendency to give away a proton, and you've got the NH3 plus that has a tendency to give away a proton as well. Which one is the best at giving away protons? Which is the most acidic? Well, it's by far and away this one. This will lose the proton first. The Ka for carboxylic acid is usually about 10 to the minus 5. In this case, it's lower than that. It's 10 to the minus 3 or something. Whereas the Ka for that is more like 10 to the minus 9. So this will give away its proton way before the NH2 group. So you can never ever get you can never get this form. That's totally impossible, where you give the NH2 unionized and the COOH unionized. Okay, we'll have a look at that in this bit here now. Okay, so what I've done here is there is the zwitterion. Now let's see what would happen if we made the pH more alkaline. Okay, so more alkaline, so increase pH. In other words, you've got to decrease the hydrogen ion concentration by adding alkali. Well, what will happen is you'll lose that hydrogen. Okay, so you will form this. That will now exist as so H2N and you've got that's still negatively charged. So that is negatively charged, so you call that's an anion. Here we call this one the zwitterion. Right, now let's decrease the pH, make it more acidic, go this way. Decrease the pH. Well, what's going to happen then, if you're going to have more acids, more H pluses about, then you're going to end up sticking a proton onto that group there. So you're going to form this species instead. So you'll have, that will be positively charged. And this will be neutral. So overall, this species will be positively charged. So it exists as the cation. And you can see at no pH do you get this species existing. That could never exist at any pH. Okay, so if I just change I'm going to put an arrow going that way, so you can obviously, these are all reversible. So if you want to go forwards, you add alkali, increase the pH. There's the red arrow. If you want to go backwards, you add acid and you decrease the pH. Now so this is the zwitterion. That is the form that predominates at pH 7. If the pH drops below about 3, so pH is less than about 3, then you're going to get the cation predominating. And if the pH is greater than about 9, you'll get the anion forming there. And this is a very common question. They'll ask you to draw the form of an amino acid that would exist at a certain pH. Usually they say at neutral pH. Right, let's just have a quick look at this table down here. Right, this is on your data sheet. This is on the AQA data sheet. Right, right. So you can see then, try and make them a bit bigger. Right, so they've given you they give you those amino acids. So just to see what is the R group in each case, and it's CH3 in alanine. In phenyl alanine, you've got a benzene ring attached to a CH2. In serine, you've got an alcohol, so you've got a third functional group in that particular amino acid. In cysteine, you've got an SH group, and we'll talk about that in a later video as well. I want to talk about aspartic acid here. So this has actually got another carboxylic acid group in it. And a very common question, it will say draw aspartic acid at pH 7. So we know that's going to exist as a zwitterion. So you would draw, let's draw it, you draw. Right, this is the carboxylic acid, so that's going to be negatively charged because it's pH 7. The amine group is going to be positively charged because it's pH 7. We've got this zwitterion form there. But we've also got this side on the R group, and that contains a carboxylic acid. And a lot of people think, oh, that one won't be ionized, that will be that. Well, it will. It will just behave exactly the same as the other carboxylic acid. At pH 7, it won't exist like that. It will be the negative ion. Okay. And the same goes for lysine as well. Lysine has got an NH2 group. So at neutral pH, that NH2 group is going to be positively charged. It's going to be NH3+. It behaves just as this NH2 group here. It's no different. So just a reminder of that. Anyway, we'll talk more about those different R groups in the next video.